My name is Chris Sibley. I'm a group leader at the University of Edinburgh and the School of Biology there. And my research focus and the team who work with me, we're focused on looking at RNA biology, so a type of biomolecule which is found in all types of cells. And we're specifically interested in how it's chopped and changed and misregulated in the context of various different neurological diseases, including motor neuron disease. So we're interested and fascinated by cause and consequence of these changes and how they link to the early stages of the disease process. So this project's all about a particular protein, which is it changes its location in the cells which are affected in motor neuron disease, and it clumps into all aggregates. And this particular protein has this kind of response in MND cases in about 98% of people who have the disease. And it's obviously therefore gained a lot of interest in the field and a lot of attention. There's been suggestion that it's possibly a bystander and a consequence of a disease. Others have thought that it's kind of involved. And I think the growing evidence is that it is implicated. Whether it's the cause of disease or not is still under debate, but certainly when this happens, it seems to be detrimental and causes a series of events which contribute to the disease etiology. So we are really interested in less about the consequences of this, but what are the molecular triggers of this event? What causes this particular protein to move from where it should be in the cell to somewhere it shouldn't be, and why does it start to aggravate? And then what we really want to do in this award is ask whether this event can be corrected, or whether it can be reversed, whether it could be used as a novel, like preventing mislocalization and kind of clumping can actually be used as a novel treatment. So as kind of context to our project, we did this really systematic study of a large number of motor neuron disease relevant models. And we identified a potentially novel translatable target, a therapeutic target, which we think might be involved in this process. So we feel that it's really pivotal that there is research which is funded into the very underlying basic molecular mechanisms of a disease, because it will allow us to understand that disease process in far greater detail, but it will also present really rationalized opportunities to target these early molecular events with new therapies which may then have translational potential. I think one of the main goals of research is trying to validate this particular candidate in causing the aggregation of this protein and mislocalization of this protein called TDP-43. So we want to validate our molecular mechanism, and that is then with a view of this potentially being a translatable target which can be pursued in future in further studies. But I guess more broadly, I hope the project also offers hope to people who have got MND, but there's people really working at this basic level, at the molecular underpinnings of the disease, trying to make a difference, trying to understand the disease and trying to develop rationalized targets and therapies based on these discoveries. First and foremost, we hope that the immediate impact is greater disease understanding. It's an unanswered question why this particular protein mislocalizes and aggregates. What are the triggers of that remains uncertain. It also remains unanswered why the disease is so rapidly progressing. So we hope that some of the findings from our study might kind of give us some insights to this. In terms of the next steps based on our findings, if all goes to plan in the project, then we hope that we validate this as a potential target for novel therapies in the future. And there's a couple of different ways in which this type of event can be corrected, and we'll be pursuing this in future applications for grant funding. I'd just like to thank the Doddie Foundation for funding research. We're all very excited here in the group and in the university. So thank you for all the supporters out there, and for all the donations which are made, which allow this kind of research to progress.